This video will continue on with information from Chapter 4, Module 1, looking at the different ways that genes can change. So by the end of the video, you should understand some of these basic different ways that genes can change, and also be able to explain how epigenetic changes might influence behaviors. So the question I'm posing to you is, how do genes change? So think back to maybe high school biology or college biology, and it's probably the term or the word that's most commonly associated or frequently thought of when we think of genetic change, and that's mutation. So mutation can cause cancer, mutation can cause different inherited disorders. It can happen in utero and cause deformities. I mean, a variety of different things. And a mutation in terms of genes is basically considered a heritable change in the DNA molecule, and typically it's just one base change. It could be more, but one base change can give you a different amino acid and protein, and that can alter the structure and the function of that protein. One example is malaria resistance. So in humans, a hemoglobin mutation makes red blood cells take on a curved sickle cell-like shape. And when you have one copy of this gene, it gives you resistance to malaria, which is something that helps our species survive. But when you have two copies, it causes sickle cell anemia. So that's an example of a heritable change that can be beneficial in one case and not beneficial in another. Some things maybe we're less familiar with might be microduplication. So microduplication is where a microscopic section of the DNA is duplicated, and it can lead to a variety of different disorders as well, one including Charcot-Marie-Tooth disease, or CMT. Perhaps microdeletion, also one we're less commonly aware of. And this is like it sounds as well, where you have a microscopic portion of the DNA that is deleted. And it can also lead to a variety of different disorders. Many are extremely rare. Another way that genes can change is called epigenetics. So epigenetics is a field that's concerned with changes in gene expression where you don't have modification of the DNA sequence. So on the previous slide, we were talking about microduplications, microdeletions, mutations. Those all involve modifications of the DNA. And so you have a change in gene expression because of a change in that actual DNA. Epigenetics is where, for some reason, we have a change in gene expression, but there is no modification of the DNA sequence. And with epigenetics comes this idea that what you do at any moment not only affects you now, but can produce epigenetic effects that alter gene expression for a longer period of time, and perhaps can be inherited by your offspring. So some examples of epigenetic changes are methylation and acetylation. So if we look at this diagram here, on the top part, we see an example of methylation. So methylation keeps the DNA, so this purple strand is the DNA, and the blue is the histone. Blue balls are the histones. And so methylation, which we see here is an added methyl group, that keeps that DNA tightly wound around the histones. And when it's tightly wound around the histones, it's not as easy to transcribe. So gene expression is going to be decreased. Acetylation, on the other hand, and here you see an acetyl group. So again, see the acetyl group's attaching to the DNA. It's not modifying the DNA sequence. So when we have an acetyl group attached, you can see that now the DNA has become unwound and it's much looser around that histone, making it much easier to transcribe this gene sequence. So that would increase that gene expression for something that was acetylated. So we'll see that these changes in transcription can result in changes in protein expression that can alter both physical and behavioral expression characteristics. So what scientists have learned about these epigenetics is that experiences can alter the activity of genes. Changes in gene expression are central to learning and memory. In addition, they could be explanations for why monozygotic twins, which are identical twins, have differences. So two researchers, Zeff and Meany, they met in a bar during a conference and set out together to design a set of experiments to look at how epigenetics might explain behavior. So they looked at rat pups. So here's a mother rat, and here's some rat pups. Here's another mother rat, and here are her rat pups. And so they looked at rat pups born to an inattentive mother. So this is bad mothering. So The bad mothering or the inattentive rat mother doesn't lick her rat pups, doesn't groom them. There's no tactile stimulation. Whereas an attentive or good mothering rat, she licks her rat pups, gives them attention. And what they found when they compared the inattentive, the pups of inattentive mother to the pups of an attentive mother or a good mother, was that there was differences in the DNA for stress-related hormones. And those stress-related hormones were more methylated in the pups bred by the bad mother or the inattentive mother. As a result, they didn't have a normal stress response, and they became nervous rats as adults. In a second experiment, they swapped the rat pups. So they gave the rat pups born to the good mother or the attentive mother to the inattentive bad mother, and they gave the pups born to the inattentive mother to the good mother. And they saw the same amount of gene expression. So even though these rat pups had been born to a good mother because they were raised by an inattentive mother or a bad mother, they still had this methylation of those stress genes. And they went on and they did some further studies looking at how this could be reversed. So the idea is that epigenetic changes occur, but they can be reversed. Since those rat studies, these epigenetic changes have also been looked at in humans in a variety of stress-induced situations. So one study that went a little step further than looking at just how parenting can alter DNA, but also how a mother who might be exposed to some sort of stressor, and looked at the DNA of the pregnant women. And so here, this is an example of a study looking at the Tutsi genocide. And so here you had pregnant women who witnessed the Tutsi genocide, and so they were experiencing PTSD at a later time, or post-traumatic stress disorder, and they compared both their DNA and the DNA of their children when they were born with mothers who didn't witness the genocide. And what they found when they compared the DNA was that those who were not exposed to the genocide and those who were had differences in the expression of stress hormones, with an increase in methylation of these genes in both the mothers and the children that witnessed the genocide, so those that had the stressor. So these findings suggest that children can inherit alterations in stress response due to things that their mothers have experienced. Now again, that's kind of a little scary maybe, but remember that this is all reversible, okay? So that those changes, that methylation or that acetylation, can be altered later on. So it's important to remember about epigenetics that it is heritable. So we just discussed how it can be passed down from the mother to the child. It can be self-perpetuating, but it is also reversible. So that's the important thing to remember. And it seems to be influenced by environment and lifestyle. So the things you do now could affect your children. So one last topic to continue, or to finish up this video, is just a little bit about heredity and talking about heritability. So the question is how much do differences in characteristics depend on these genetic differences, no matter how they occur? So researchers have found evidence for heritability in almost every behavior they've tested. Just to make sure we all understand what heritability is, heritability is the variation in a given trait that can be attributed to genetic differences within a population. So say we have a trait like intelligence, and we can all agree that everybody has differing levels of intelligence. So how much of those differences in intelligence can be attributed to genetic differences? Typically, heritability is studied with twins and adoption studies comparing identical twins to fraternal twins to regular siblings, comparing biological parents and children to adoptive parents and children, and looking at behaviors and seeing how similar those are. Newer evidence actually links specific genes to behaviors. So now that we have the human genome map, we can actually look and see, are there specific genes that are associated with behaviors? And it's important to understand that heritability is often overestimated just because of the ways that we study it. But this, looking at specific genes linked to behavior, is much more promising.